Today we are going to discuss the structure and function of Fas, the cell death receptor. Fas is a 335 amino acid type 1 transmembrane glycoprotein located on the cell surface. The receptor and its ligand, Fas-L, are both membrane-bound homotrimers. Fas is generated from the CD95 gene and is part of the TNF receptor superfamily. The receptor is composed of three cysteine-rich domains: the N-terminal ligand-binding extracellular domain, the transmembrane domain, and the C-terminal cytoplasmic death domain, which we will focus on.
The death domain is critical for apoptotic signaling. It serves for protein-protein interactions, and it allows for recruitment of signaling proteins. This is the extrinsic pathway of apoptosis in the immune system, which we will use to show how Fas functions. The trimeric membrane protein Fas-L, or the Fas ligand, on the killer lymphocyte binds to a Fas receptor on the target cell surface. Binding of the membrane-bound Fas-L to the Fas receptor triggers receptor clustering and oligomerization, which causes activation of the death domains on the C-terminal tails of the receptors. Activated Fas receptors cause Fad adapter proteins to attach to receptor tails via death domains. Fad adapters recruit caspase 8 proteins, which are attached via death effector domains, or DEDs. With all these proteins coming together, you get the formation of the disc complex. Within the disc, there are two adjacent caspase 8 molecules that will cleave one another to create an active protease dimer. This dimer activates itself from disc at its death effector domain, which now makes it a soluble and active caspase 8 enzyme.
Active caspase 8 then targets downstream executioner caspases, which are additional aspartate-specific cysteine caspases in the cell that lead to cleavage of structural and regulatory proteins. And from this signaling, you get apoptosis of the target cell. Fas-L's configuration can actually change the fate of the cell. So when Fas-L is membrane-bound, this state is known as M-Fas-L. It is able to bind to the Fas receptor and induce apoptosis. This is shown in the extrinsic apoptotic pathway that we just discussed. However, Fas-L can also be cleaved, which results in a change of the cell's fate. So if Fas-L is cleaved by a metalloprotease, it becomes soluble, and this form is known as S-Fas-L, S standing for soluble. In this state, S-Fas-L is unable to trigger receptor oligomerization, and thus there's a failure to induce apoptosis. And when S-Fas-L is created, it is able to induce proliferative signals. Then when S-Fas-L binds to the Fas receptor, it now has the ability to inhibit the M-Fas-L apoptotic pathway. And this inhibition occurs because S-Fas-L binds to the Fas receptor via steric hindrance. S-Fas-L binding promotes proliferative activity by signaling for proteins known as ERK, PI3K, and one called JNK. And activity of these proteins inhibits apoptosis and results actually in cell survival.